Okay, thank you. First of all, I want to thank the organizers for their opportunity they provided me to talk to you about our research. And I particularly want to say I look forward to seeing all in person at the next SRCA symposium. Hopefully, this will be a typical in-person meeting. So today I'm going to talk to you about our work on the cholecystokinin 1 receptor activation and how it restores normal mTOR C1 signaling and is protective to Purkinje cells in a couple of SCA mouse models. I have no disclosures to claim at this point, so let's proceed with the talk. First of all, it's important to note that there are several dominant SCAs that are caused by CAG expansions, and in particular, it's, for example, SCAs 1, 2, 3, 6, 7, and 17. And these typically show rather substantial atrophy of Purkinje cells in the cerebellum. And what I'm showing here is an MRI of an individual affected with SCA1, and you can see the atrophy in this individual's cerebellum. Upon death and subsequent autopsy, you can note the loss of Purkinje cells from the cerebellar cortex, as well as the atrophy of the dendritic tree and the few Purkinje cells remaining in this individual's cerebellum. And again, back to an earlier point I raised, the importance of Purkinje cell pathology in the SCAs, in particular SCA1 and 2 and SCA6 show very prominent and consistent degeneration of Purkinje cells, whereas in SCA3, perhaps the most prevalent SCA disorder, the Purkinje cell atrophy is much milder and more variable. But once we cloned the SCA1 gene and the importance of Purkinje cell pathology in this disease, we decided to develop a strategy where we could begin to look at pathogenesis specifically in Purkinje cells due to an expanded ataxin-1 protein. And so we used a classic transgenic approach using the PCP2L7 promoter to drive expression of various versions of ataxin-1, specifically in Purkinje cells of the cerebellum in mice. And in this case, I'm showing you the results of when we expressed in a ataxin-1 with 82 glutamines. And this shows you the ability of these regulatory region to target the Purkinje cells specifically. These mice develop ataxia. More recently, it's been shown that Purkinje cells of these mice show a very classic reduction in the rate of their spontaneous firing. And if we do a sort of similar MRI image analysis on these cerebellum of these mice, as I showed earlier in the SCA1 patients, you can see at 1 year the dramatic atrophy of the cerebellum in these mice. And this is correlated with a loss of Purkinje cells from the cerebellar cortex as indicated by calbindin staining. So I think this provides evidence that at least in terms of motor phenotype and pathology, this model nicely replicates what we see in the humans SCA1 patients. So over the years, as they say, we used this strategy to, if you will, to kind of molecularly dissect the regions of the ataxin-1 protein that are important for pathogenesis in Purkinje cells. And in addition to the expanded glutamine tract, the AXH region, which forms an OB fold, interacts with many nuclear proteins and most notably Capicua, and this interaction is important for driving pathogenesis. Further towards the C-terminus is the nuclear localization sequence of the ataxin-1 protein, and we showed that entry of this protein into the nucleus of Purkinje cells is important to disease. But what I want to focus on now is the work we did examining the phosphorylation of serine at position 776. And I should say the nomenclature for amino acid position numbers is based on a protein with 30 glutamines. So if we make these transgenic mice with an alanine at position 776 with an 82 glutamine ataxin 1, so that this is a residue that can't be phosphorylated, the disease in this, the severity of disease in these mice is very dramatically reduced. In contrast, if we put in a phosphomimetic amino acid at this position and aspartic acid, we get a dramatic increase in severity of disease, Purkinje cell disease. We went on then to test what happens if we take a wild type version of the ataxin-1 protein with 30 glutamines, express it in Purkinje cells, and in this case, we have a phosphomimetic version. And the really interesting thing is that these animals are just as ataxic as the animals expressing the expanded version of 82 glutamines, but they show a dramatic reduction in the degeneration of Purkinje cells, most notably as assessed by the thickness of the molecular layer of the cerebellar cortex. You can see that the atrophy of Purkinje cells is dramatically reduced in these 30Q D776 mice. And this raised the very intriguing question that perhaps in these Purkinje cells of the 30Q D776 mice, there was some protective pathway that was activated compared to Purkinje cells in the cerebellum of the ataxin-1 82Q mice. And so we used RNA-seq to begin to get at this question. And by doing a number of comparisons in 12-week-old mice, we found 130 genes were different in all genotypes that we compared. And what we focused on were those genes that were up in the 32Q mice with the D776 compared to those that were down in the ataxin-1 82Q. So the hypothesis is we've activated a protective pathway and we should see enhanced expression of key genes in this pathway. We went through this set of 32 genes and picked out those that were Purkinje cell specific and had a reasonable level of expression. And what we found were two genes. One was CCK, which is cholecystokinin, and another is an isoform of collagen. Given that the effect of CCK expression was much more dramatic in the D30 mice, we started looking at whether CCK might be an entryway into understanding why pathogenesis of Purkinje cells was so dramatically reduced in these D30 mice. So just a little background, CCK is a well-known pro-peptide hormone that is really classically associated with aspects of the gastrointestinal system, including a gallbladder contraction and gastric emptying, and in the CNS, the induction of satiation and suppression of food intake. The functional elements of this protein are peptides that are generated by cleavage, indicated here, and most notably, for example, is CCK8. And these all interact with the CCK1 receptor. This cholecystokinin is a member of the gastrin family, whereas the gastrin-generated peptides typically favor interaction with the CCK2 receptor. Because of the role of this system in satiation and potentially obesity, there has been an enormous amount of effort to try to identify small molecules that might stimulate these two receptors as potential therapeutics. So first, let's move to see if we can get a little clearer picture of the role of the CCK system in the D30 mice in terms of the absence of Purkinje cell atrophy. And again, here we're looking at molecular layer thickness in 6-week versus 1 8-year-old animals, and basically absence of atrophy in the 30Q D776 mice compared to significant molecular layer atrophy in the 82Q mice. If we now cross our D30 mice with mice that lack CCK, which is very robustly expressed in Purkinje cells, you now see that we've generated a condition very similar to the ataxin-1 82Q mice with significant atrophy of the Purkinje cell of the molecular layer thickness and Purkinje cell dendrites. If we now cross the D30 mice to knockout mice for either the CCK1 receptor or the CCK2 receptor, we see, similar to the CCK1 knockout mice, D30s on this background show progressive Purkinje cell atrophy, whereas the D30 transgene on the CCK2 knockout background do not show atrophy of the molecular layer thickness or Purkinje cells. And appropriately, CCK1 receptor is expressed in the dendritic tree of the cerebellum, in the Purkinje cells, excuse me, of the cerebellum, whereas CCK2 receptor is not. So to explore the role of the CCK1 receptor in Purkinje cell atrophy, we went to a small molecule called A71623, and this is a peptide mimetic, if you will. And as you can see, it's very selective over the CCK1 receptor versus the CCK2, something on the order of 1,000-fold. And the first experiment we did was to assess whether by IP injection we could stimulate the CCK1 receptor. And a classic way of doing this is looking at the extracellular signaling regulator or kinase phosphorylation. And what we found was that we could get ERK phosphorylation by IP injection, and this was a dose responsive effect of, ERK phosphorylation in the cerebellum. We then went on to see if administration of this small molecule would impact the disease progression in our 30Q D776 mice on the CCK knockout background. And this shown at the top is the scheme that we did. We placed IP pumps that were replaced. And then at the end, we did both a behavioral as well as pathology in the mice. And importantly, by rotarod, here is the loss of rotarod performance in vehicle-treated mice. And you can see this loss of performance is dramatically diminished in those mice that receive the agonist. And correspondingly, the atrophy of the molecular layer in mice receiving the vehicle was protected in mice receiving the agonist. So in this system, the activation of the CCK1 receptor seemed to give us a protective effect against the disease. We then moved on to do a similar experiment in the 82 ataxin-1 82 transgenic mice using basically the same paradigm. We did add the beam cross to our motor performance. And here you can see that in vehicle-treated animals at 11 weeks versus 3 weeks, the increase in deficit, if you will, in motor performance by an increase in time to cross or foot slips on the balance beam. Animals receiving the agonist did not show this deficit in motor performance. And similarly, animals receiving the agonist, the 82Q animals, did not show the deficit in motor performance as assessed by rotarod. And finally, in the 82Q mice, the agonist protected animals against the atrophy of the Purkinje cell dendrites as assessed by molecular layer thickness. So in 2 SEA1 models, if you will, this agonist proved to be effective in terms of disease phenotypes. Now, if we look at what sort of pathways are stimulated by the CCK1 receptor, one pathway of note is the mTOR pathway. And we're particularly intrigued with this pathway because mTOR C1 expression has shown to be impaired in another mouse model of SCA1, the 154 knock-in mice. And so what we did was to look at the mTOR signaling pathway in the animals receiving the agonist. And specifically, we're looking at the ability of mTOR C1 to stimulate the kinase that phosphorylates the ribosomal protein S6. And so that will be the focus of the next series of studies. And so here is the results we got in treating our 82Q mice by IP injection of the agonist A71623 at 11 weeks of age, and the assessment of protein phosphorylation and protein levels at 24 hours after the IP injection. And it's a bit complicated, but these are the wild type and wild type vehicle and wild type receiving agonist mice compared to the 82Q mice receiving vehicle. And you can now see a reduction in phosphorylation of this ribosomal protein. And in those 82Q animals receiving the agonist, this phosphorylation of this ribosomal protein is increased with the data shown down here quantitatively. And this is a significant increase. Interestingly, we also see a slight but significant increase in calbindin in the 82Q animals given the agonist. Importantly, if we do this same experiment in our 82Q animals that have been crossed onto the CCK1 receptor knockout background, we do not see an increase in phosphorylation of the S6 ribosomal protein. So again, the ability of this compound to act is dependent on the presence of the CCK1 receptor. Next, we moved on to another SCA1 mouse model, the ataxin 2 127 transgenic, 127 glutamine transgenic animals generated in Stefan Pulse Lab. And in our hands, these animals only showed a balance beam phenotype. And the bottom line is that administration of the CCK1 agonist had the same protective effect on the motor performance in these animals as we saw in our SCA1 mouse models. So again, looking at the 3 versus 11 weeks of age, you can see an improvement both in terms of foot slips as well as time to cross, significant improvement compared to the animals receiving vehicle, such that their performance is now not significantly different from wild type animals, both in terms of foot slips or time to cross. Moving to the phosphorylation of the ribosomal protein, again, by Western blot analysis using the same sort of paradigm that we went through previously with the 82Q ataxin 1 mice. The interesting thing is that these animals, the ataxin 2 transgenic animals, rather than showing a deficit in mTOR signaling as indicated by S6 phosphorylation, they show an enhanced signaling of the mTOR C1 pathway compared to wild type animals. And importantly, administering the agonist at various doses reduced the signaling level back down to the wild type levels, as shown quantitatively over here. And importantly, as in the 82Q animals, ataxin 1 animals, we saw a slight but significant increase in calbindin in those animals, the SCA2 animals receiving agonist. Now, very interestingly, it has been shown in this paper that looking at mTOR C1 and mTOR-C2 function in Purkinje cells, they reported a convergence between the phenotypes, i.e. degeneration of Purkinje cells lacking mTOR activity, as we saw in our SEA1 models, and those expressing constitutively active mTOR-C1, as we saw in our SEA2 models. So the idea that is developing is that while mTOR signaling activity is very important for Purkinje cell function, you can either have too little or too much to get adverse effects. So in summary, our results indicate that in Purkinje cells, there's this pathway that involves CCK cleavage to CCK8, we speculate, and then subsequent autocrine activity of the CCK1 receptor and through the mTOR signaling, enabling the cell to respond more effectively to various stressful conditions, such as polyglutamine expanded ataxin 1 or ataxin 2. And so basically in our ataxin 1 mice, we see a decrease in mTOR signaling. In our ataxin 2 mice, there is an increase in mTOR signaling, but importantly, in both instances, the CCK1 receptor activation restores signaling to normal levels. In closing, we've just recently found that in the 154 SEA1 model, the knock-in mice, there's a reduction in CCK expression, not only in the cerebellum, but other regions affected by expression of the mutant ataxin 1 protein in the cortex, hippocampus, and striatum. And interestingly, it was reported several years ago that restoring mTOR signaling activity in a Huntington's disease mouse model improves the disease phenotype. So we're intrigued with the idea that the CCK1 CCK and CCK1 receptor pathway may have broader implications in different regions in other neurodegenerative diseases. So I thank you for your attention and point out the members of our group here in Minnesota who played important contributions, as well as our collaborators here. And importantly, this work was done in collaboration in terms of the SEA2 analysis with Stefan Pulst, where Sharon Paul and Carla Figueroa were important contributors. So again, thank you very much for your attention.